Hey guys, it's Professor Beda. Let's learn about membrane structure and function. We probably know that living organisms are made up of cells from unicellular organisms like bacteria, which are made up of just one cell, to large animals that are made up of trillion of cells. One key feature of a cell is the cell membrane or plasma membrane, which separates what's inside the cell from what's outside the cell. This is a very important function because an organism must be distinct from its surrounding. Inside the cell is where metabolism takes place and genetic information is stored under regulated conditions or homeostasis. The cell membrane is semi-permeable, meaning it lets something through while preventing other things. And now that we know about lipids with chapter 5, the macromolecule, so we are ready to understand the outermost layer of the cell in more details. Before the advent of electron microscope, some biologists and researchers identified cell membranes using indirect evidence and hypothesized different models which gave us the basic idea of plasma membrane structure. In 1925, Gorter and Grindel stated that plasma membrane is made up of double layer of lipid molecules. They put the foundation of future models of membrane structure. And in 1935, Davzan and Danieli model, they proposed their model, a sandwich model, protein, lipid bilayer, and protein, uh, which included the phospholipid bilayer on the inside of, uh, of their model and of the cell membrane, and a globular protein coating both sides or of the outer surface of the cell membrane. And then we have the unit membrane model was proposed by Robertson in 1959, and it's similar to the sandwich model of Davzin and Danieli, except that in Robertson model, the protein type is beta fibrous protein. That means the protein is fibrous, not globular, as in Davzin Danieli model. Now we have the most universally acceptable model, the fluid mosaic model. It was proposed by Singer and Nicholson in 1972. According to this model, lipid and intrinsic protein are present in mosaic arrangement, and the biological membrane are semi-fluid so that lipid as well as intrinsic protein are able to make movement within the lipid bilayer. Lipids and proteins are the staple ingredients of membranes, although carbohydrates are also important. The most abundant lipids in most membranes are phospholipids. A phospholipid is an amphipathic molecule, meaning it has both hydrophilic water-loving region and hydrophobic water-fearing region. A phospholipid can exist a stable boundary between two aqueous compartments because the molecular arrangement shelters the hydrophobic uh, tails of the phospholipid from water while exposing the hydrophilic head to water. This molecular orientation maximizes contact of hydrophilic regions of molecules. Um, the hydrophilic region is away from water and within with the cytosol and extracellular fluid while providing the hydrophobic parts with non-aqueous environment. This image and we have this image shows the currently acceptable or accepted model of arrangement of molecules in plasma membrane. In this fluid mosaic model, the membrane is a mosaic of protein molecules bobbing in fluid bilayer of phospholipids. The proteins are not randomly distributed in the membrane. Groups of proteins are often associated in long-lasting specialized batches where they carry out a common function, Like all models, the fluid mosaic model is continually being refined as new research reveal more about membrane structure. Membrane are not static sheets of molecules locked rigidly in place. A membrane is held together by hydrophobic interactions, which are much weaker than covalent bonds. Most of the lipids and some proteins can shift about sideways in the plane of the membrane, and very rarely a lipid may flip-flop across the membrane, switching from one phospholipid layer to the other. This occurs rapidly, just a few parts of a second. Proteins are much larger than lipids and move more slowly. Many membrane proteins seem to be held immobile by their attachment to the cytoskeleton or to the extracellular matrix. In this experiment, Larry and Michael at Johns Hopkins University labeled the plasma membrane proteins of mouse and human cells with two different markers, and then they fused them. Those markers may be isotopes or fluorescence dyes, special dyes. Uh, they they uh, they labeled these two cells, mouse and human. If you appear here with pink and blue colors, they label them with two different markers, and then they fuse the cell after 1 hour. Using a microscope, they observe the markers, both of them, appears on the hybrid cells. They conclude that the mixing of mouse and human membrane proteins indicate that at least some membrane proteins move sideways within the plane of the plasma membrane. A membrane remains fluid as temperature decreases until the phospholipids settle into a closely packed arrangement and the membrane solidifies, much as a baking grease forms lard when it cools. As the temperature decreases, the membrane remains fluid to a lower temperature. If it is rich in phospholipids with unsaturated hydrocarbon tails. Uh, because of the kink in the tails where double bonds are located, remember that they're double bond in the hydrocarbon tails of fatty acids, they will form kink in the hydrocarbon tail. So unsaturated hydrocarbon tail cannot pack together or closely as saturated hydrocarbon tails, making the membrane more fluid. So here in this side on the left, unsaturated hydrocarbon tails kinked. You see this kinked in the fatty acid cells, prevent packing in the the packing of the phospholipid together, closely packing together, enhancing membrane fluidity. And here, saturated hydrocarbon tails pack together, increasing membrane viscosity. The steroids, which is wedged between a phospholipid molecule and the plasma membrane of animal cells, has a different effect on membrane fluidity at different temperatures. At 37 Celsius, the body temperature of a human, cholesterol makes the membrane less fluid by restraining phospholipid movement. However, because cholesterol also hinders the close packing of phospholipid, it lowers the temperature required for the membrane to solidify. Thus, cholesterol can be thought of as a fluidity buffer for the membrane, resisting changes in membrane fluidity that can be caused by changes in temperature. Compared to animal cells, plants have very low levels of cholesterol. Rather, related steroid lipids buffer membrane fluidity in plant cells. Membrane must be fluid to work properly. The fluidity of membrane affects both its permeability and the ability of membrane proteins to move to where their function is required or needed. Usually, membrane are about as fluid as olive oil. When membrane solidifies, its permeability changes and enzymatic protein in the membrane may become inactive. if their activity require movement within the membrane. The fluid mosaic model is somewhat like a tile mosaic shown here. A membrane is a collage of different proteins, often clustered together in groups, embedded in the fluid matrix of lipid bilayer. A phospholipids form the main fabric. See those phospholipids, they form the main fabric of the membrane, but proteins determine most of the membrane's function. Different types of cells contain different sets of membrane proteins, and uh, various membranes within a cell each have a unique collection of proteins. Note that in the upper image that there are two major populations of membrane proteins, integral proteins and peripheral proteins. Integral proteins penetrate the hydrophobic interior of the membrane or for the lipid bilayer. The majority are transmembrane proteins which span the membrane. Other integral proteins extend only partway into the hydrophobic interior. The hydrophobic region of an integral protein consists of one or more stretches of nonpolar amino acids. We have 20 amino acids, and if you remember them, we have 10 or 12 of them. They are nonpolar. So nonpolar amino acids form the hydrophobic region of transmembrane proteins. 20 or 30 amino acids in length, usually coiled into alpha helix. The hydrophilic parts of a molecule are exposed to the aqueous solution on either sides of the membrane. Some proteins also have one or more hydrophilic channels passage through the membrane of hydrophilic substances. Peripheral proteins are not embedded in the lipid bilayer at all. They are loosely bonded to the surface of the membrane, often to expose part of integral protein. On the cytoplasmic side of the plasma membrane, some membrane protein are held in place by attachment to the cytoskeleton, and on the extracellular side, certain membrane proteins may attach to material outside the cell. They may attach to fibers of the extracellular matrix, the collagen and fibronectin. Six major functions performed by proteins of the plasma membrane. Thus, the membrane is a functional mosaic as well as structural mosaic. Transports protein function as or serve as transport. On the left, a protein that spans the membrane may provide hydrophilic channels across the membrane that is selective for particular solutes. On the right, other transport proteins shuttle a substance from one side to other by changing shape. Some of these proteins hydrolyze ATP as a source of energy to actively pump substances across the membrane. And we have enzymatic activity for protein. A protein built into the membrane may be an enzyme with its active sites. Those active sites where the reactant binds, exposed to substance in the adjacent solution. In some cases, several enzymes in a membrane are organized as a team that carries out sequential steps of metabolic pathways. We have a good example or obvious example in the inner membrane of the mitochondria. It contains a lot of those teams or enzymes teams that performs sequential steps in the cellular respiration. And then we have or proteins serve as signal transduction. Uh, we call those type of protein receptor. May have a binding site with a specific shape that fits the shape of chemical messenger, those such as a hormone. The external messenger signalic molecule may cause the protein to change shape. When they change shape, they allowing it to relay the message to the inside of the cell, usually by binding to a cytoplasmic protein. For example, insulin, the hormone insulin, it's secreted from the beta cells from the pancreas. They send or they secrete insulin when the blood glucose concentration raises, and the insulin bring a message from pancreas, tell the cell to open the gate to glucose because their concentration in the blood is too high. Cell-to-cell recognition, some glycoproteins serve as identification tags um, that are specifically recognized by membrane proteins of other cells. This is the proteins or membrane protein of other cells. This type of cell-to-cell binding is usually short-lived, and in intracellular joining, membrane proteins of adjacent cells may hook together in various kinds of junctions, such as gap junctions or tight junctions. This is long-lasting binding. Uh, in this attachment of the cytoskeleton, those protein to cytoskeleton or extracellular matrix, uh, microfilament of or other elements of the cytoskeleton may be non-covalently bound to membrane proteins, a function that helps maintain cell shape and stabilizes the location of certain membrane proteins. Uh, a cell's ability to distinguish one type of neighboring cell from another is this is cell-to-cell recognition, and is crucial to functioning of an organism. It's important, for example, in sorting cells into tissue and organs in animal embryo. It is also the basis for rejection of foreign cells by the immune system and an important line of defense in vertebrate animals. Membrane carbohydrates are covalently bonded to lipids, forming molecules called glycolipids and glycoamine carbohydrates. And most are covalently bonded to proteins, and they form glycoproteins. Keep in mind that carbohydrates on the extracellular side of the plasma membrane vary those the green hexagonal shape. Those are carbohydrates, sugars. Each one is monosaccharides. Remember that they form carbohydrates, the macromolecule, and those carbohydrates on extracellular side, they form the glycoproteins when they attach to protein. Those vary from species to species among individuals of the same species. So they vary from species to species and even among individuals of the same species. And they are varies from one cell type to another in a single individual. This functions as markers that distinguish one cell from another. We have an example in human blood type designated to A, B, AB, and O. Those variation in human blood types reflect variation in carbohydrate part of the glycoprotein on the surface of red blood cells. The biological membrane has emergent properties beyond those of many individual molecules that make it up. We will focus on selective transport or selective permeability. That is it allows some substances to cross more easily than others. We will see once again that fluid mosaic model helps explain how membranes regulate the cell's molecular traffic. So, a steady traffic of small molecules and ions moves across the plasma membrane in both directions. For example, consider the chemical exchanges between a muscle cell and extracellular fluid that passes it. Sugars, amino acids, and other nutrients enter the cell, and metabolic waste products leave it. The cell takes an oxygen for use in cellular respiration and expel carbon dioxide. Also, the cell regulates its concentration of inorganic ions such as sodium, potassium, calcium, and chloride by shuttling them one way or other across the plasma membrane. In spite of heavy traffic through them, cell membrane and cell membrane are selective in their permeability. Substances don't cross the barrier indiscriminately. The cell is able to take uh, or to take up some small molecules and ions and exclude others. Nonpolar molecules such as hydrocarbons, carbon dioxide, and oxygen are hydrophobic, as are lipids. They can all, therefore, dissolve in lipid bilayer of the membrane and cross it easily without the aid of membrane proteins. However, the hydrophobic interior of the plasma membrane impedes direct passage through the membrane of ions and polar molecules, which are hydrophilic. Polar molecules doesn't cross rapidly relative to nonpolar molecules. The lipid bilayer is only one aspect of the gatekeeper system responsible for cells' selective permeability. Proteins built into the membrane play key roles in regulating transport. Molecules have a type of energy we call the kinetic energy, if you remember those kinetic energy, and they have a thermal energy due to their constant motion. One result of this motion is diffusion. Diffusion is the movement of particles of any substances um, so that they spread out into the available space. Each molecule moves randomly, even though diffusion of a population of molecules may be directional. Molecules of a dye can pass through membrane pores. Uh, a random movement of dye molecules will cause some to pass through the pores. This happens more often on the side with more dye molecules. The dye diffuses from the more concentrated side to the less concentrated side. We call this diffusion down a concentration gradient. And then a dynamic equilibrium result. Solute molecules still cross, but at roughly equal rates in both directions. Um, Here is a simple rule of diffusion. In the absence of any other forces, a substance will diffuse from where it is more concentrated to where it is less concentrated. Okay, put another way, uh, is a substance diffuse down its concentration gradient. This is diffusion. Diffusion is a spontaneous process needing no input of energy. Each substance diffuses down its own concentration gradient, unaffected by the concentration gradient of the other substance. In this image, solution of two different dyes are separated by a membrane that is permeable to both. Each dye diffuse down its concentration gradient. There will be a net diffusion of the purple dye toward the left, even though the total solution concentration was initially greater on the left side. When a substance is more concentrated on one side of the membrane than on the other, there is a tendency for it to diffuse across down its concentration gradient, assuming that the membrane is permeable to that substance. One important example is the uptake of oxygen by a cell performing cellular respiration. The diffusion of a substance across the biological membrane is called passive transport. We call them passive transport. Because it requires no energy, the concentration gradient itself represents potential energy and drives diffusion. Remember that water can diffuse very rapidly across the membrane of cell with equiporins. The movement of water across the plasma membrane has important consequence for cells. So the movement of water across the membrane, we call it osmosis. The diffusion of free water across selectively permeable membrane, whether artificial or cellular, is called osmosis. To see how two solutions with different solute concentration interact, picture a U-shaped glass tube with selectively permeable artificial membrane separating two sugar solutions. Pores in this synthetic membrane are too small for sugar molecules to pass through, but large enough for water molecules. Okay. However, tight clustering of water molecules around the hydrophilic solute molecules makes some of water unavailable to cross the membrane. As a result, the solution with higher solutes, the concentration with the high solute concentration has lower free water concentration. So water diffuses across the membrane from the region of higher free water, here on the left, to the lower concentration, to the lower free water molecules concentration, until the solute concentration on both sides of the membrane are more nearly equal. Okay, so osmosis is the movement of water molecules across selective permeable membrane. Remember that when hydrogen or the oxygen and hydrogen molecules of water cover hydrophilic particles, what we call this molecules, okay, this zone, what we call it. We turn to the chapter of water. To explain the behavior of a cell in a solution, we must consider both the solute concentration and membrane permeability. Both factors are taken into account in the concept of tonicity. The ability of surrounding solution to cause a cell to gain or lose water. The tonicity of a solution depends in part on its concentration of solutes that cannot cross the membrane, non-penetrating solutes. They can penetrate the membrane relative to that inside the cell. Memorizing the definition of hypertonic, hypotonic, and isotonic solutions is not necessary when you understand the root words that make up these descriptive words. The difference between the words hypertonic, hypotonic, and isotonic describe the tonicity. So the tonicity is the difference in water concentration of two solutions separated by semipermeable membrane. Hypertonic is hyper mean above or excessive. Remember that, or think of hyperactive kids, which mean overly active kids. Hypertonic solution will have higher concentration of solute than the solute itself being compared to hypotonic. Hypotonic, the root hypo mean below. Think of people who are in cold temperatures that get hypothermia. The body temperature is below normal. Iso, the root of word iso mean equal. If you remember, hypertonic and hypotonic are higher and lower concentration, then isotonic must be the case where both solutions have equal concentration. Many times when talking about tonicity, examples of red blood cells in a solution are used. If there is a higher concentration of non-penetrating solutes in the surrounding solution, water will tend to leave the cell and vice versa. If a cell without cell wall, you know that animal cells, they don't have cell wall, immersed in isotonic solution, there will be no net movement of water across the plasma membrane. Water diffuses across the membrane, but at the same rate in both directions. Okay, the water will still move through the membrane, but with the same rate in both directions. An isotonic environment of the volume of an animal cell is stable. So in this isotonic solution, the volume of the cell is no change in their volume. Let's transfer the cell to the solution that is hyper. Here, hyper. Note that the cell will lose their water. So the cell will lose water, shrivel, okay, and probably die. This is why an increase in the salinity or saltness of lake can kill the animals. If the lake water becomes hypertonic to those animals. Cells, they might shrivel and die. However, taking up too much water can be just hazardous to a cell as losing water. If we place the cell in a solution that is hypotonic to the cell and hypo mean below or less, water will enter the cell faster than it leaves, and the cell will swell and lyse or burst like an overfilled water balloon. A cell without rigid cell walls can tolerate either excessive uptake or excessive use of water. This problem of water balance is automatically solved if such a cell lives in isotonic surrounding. Organisms that lack rigid cell walls must have either adaptations for osmoregulation. This is the control of solute concentrations and water balance. For example, the unicellular protist Paramecium lives in pond water, which is hypotonic to the cell. Paramecium has a plasma membrane that is much less permeable to water than the membrane of most other cells, but this only to water than the membrane of most other cells. But this only slows the uptake of water, which continually enter the cell. The reason the paramecium cell doesn't burst is that has contractile vacuole, an organelle that function as a pump to force water out of the cell as fast as it's entered by osmosis. In contrast, the bacteria and archaea that live in hypersaline, excessively salty environments have cellular mechanisms uh, that balance the internal and external solute concentration to ensure that water doesn't move out of the cell. The cell of plants, prokaryotes, fungi, and some protists are surrounded by cell wall. When such a cell is immersed in hypertonic solution, uh, but in rainwater, for example, the cell will help maintain the cell's water balance. Here, the plant cell swell and as water enter by osmosis. However, the relatively inelastic cell wall will expand only so much before it exerts a back pressure on the cell called the turgor pressure that opposes further water uptake. At this time, the cell is stirred or very firm, which is a healthy state for most plant cells. Plants that are not woody, such as most house plants, depend for mechanical support on cells kept stirred by surrounding hypotonic solution. In plant cells and the surrounding are isotonic. If you put those cells in this isotonic, there is no net tendency for water to enter and the cell become flaccid uh, or limp. The plant will wilt. In hypertonic solution or environment, a plant cell, like an animal cell, will lose water to its surrounding and shrink. As a plant cell shrinks, its plasma membrane pulls away from the cell wall at multiple places. This plasma or this phenomenon called plasmolysis causes the plant to wilt and can lead to plant death. As I mentioned before, many polar molecules and ions blocked by the lipid bilayer of the membrane diffuse passively with the help of transport proteins that span the membrane. This phenomenon is called facilitated diffusion. Most transport proteins are very specific. They transport some substances but not others. And as I mentioned before, in protein functions, the two types of transport proteins are channel protein and carrier protein. We have two types of those. Channel proteins simply provide corridors that allows specific molecules or ions to cross the membrane. The hydrophilic pathway provided by these proteins can allow water molecules or small ions to diffuse very quickly from one side of the membrane to the other. Equiporins, the water channel proteins, facilitate the massive levels of diffusion of water by osmosis that occur in plant cells and animal cells such as RBCs. Kidneys or kidney cells also have a higher number of equiporins, allowing them to reclaim water from urine before it is excreted. Channel proteins uh, that transport ions are called ion channels. Many ion channels functions as gated channels, which open or close in response to stimulus. Carrier proteins such as the glucose transporter seem to undergo a subtle change in shape that somehow translocates the solute binding site across the membrane. Such a change in shape may be triggered by the binding and release of the transported molecules. Like ion channels, carrier proteins involved in facilitated diffusion result in the net movement of substances down its concentration gradients. Despite the help of transport proteins, facilitated diffusion is considered passive transport because the solute is moving down its concentration gradient, uh, a process that requires no energy. Some other transport proteins uh, can use energy to move solute against their concentration gradients across the plasma membrane. From the side where they are less concentration or less concentrated to the side where they are more concentrated. Uh, to pump a solute across a membrane against its gradient requires work. The cell must expend energy. Therefore, this type of membrane traffic is called active transport. The transport proteins that move solutes against their concentration gradient are all carrier proteins rather than channel proteins. Active transport enables a cell to maintain internal concentrations of small solutes that differ from concentrations in its environment. The sodium potassium pump, a specific case of active transport. This transport system pumps ions against steep concentration gradients. So something like a sodium or potassium ion, when moving with the gradient from high concentration to low concentration, can pass through by passive transport, which is a kind of diffusion. But when things need to move against the concentration gradient, active transport is required. This process should be energy expenditure involved. So certain proteins like a sodium potassium pump use up ATP, the molecule that is the currency of cellular energy to move sodium ions out of the cell and potassium ions into the cell against the concentration gradient from low concentration to high concentration. I've said before that all cells have fold duct across their plasma membrane. The cytoplasmic side of the membrane is negative charge relative to the extracellular side, which is positive in charge. The ball duct across the membrane called the membrane potential. Uh, nerve A nerve impulse is an electrical signal that travels along the neuron. Uh, but it's not quite electricity as you may know it. Electricity is the flow of electrons, but in nerve impulse, we use the word potential to describe the movement of charged particles across the membrane of cell. The charged particle flow like a wave down the axon, moving in and out of the cell. Because the inside of the cell is negative compared to the outside, the membrane potential favors the passive transport of cations into the cell and anion out of the cell. Thus, two forces drive the diffusion of ions across the membrane. We have a chemical force, the ion concentration gradient, and an electrical force, the effect of the membrane potential on the ion's movement. This combination of forces acting on an ion is called the electrochemical gradient. A transport protein that generates a voltage across membrane is called an electrogenic pump. The sodium-potassium pump appears to be the major electrogenic pump of animal cells. The main electrogenic pump of plants, fungi, and bacteria is a proton pump, which actively transport protons. Protons, those are hydrogen ions, out of the cell. A solute that exists in different concentration across a membrane can do work as it moves across the membrane by diffusion down its concentration gradient. This is analogous to water that has been pumped uphill and performs work as it flows back down in a mechanism called cone transport. A carrier protein such as this hydrogen sucrose co-transporter in a plant cell, the top of this image here. This is co-transporter protein for sucrose and hydrogen ion. This one is able to use the diffusion of hydrogen ion down its electrochemical gradient into the cell to drive the uptake of sucrose, and the cell wall is not shown here, although not technically part of the transport process cell wall. An ATP driven proton pump is shown here in the bottom, which concentrates hydrogen outside the cell. The resulting hydrogen gradient represents potential energy that can be used for active transport of sucrose. In this case, the ATP hydrolysis indirectly provides the energy necessary for co-transport. So the cell uses a proton pump to actively transport hydrogen outside the cell, and the concentration gradient of hydrogen ion drive the hydrogen into inside the cell, while the co-transporter protein diffuse the hydrogen into inside the cell. It facilitates the transport of sucrose also into inside the cell. Large molecules such as proteins and polysaccharides generally don't cross the membrane by diffusion or transport proteins. Instead, they usually enter and leave the cell in bulk, packed in vesicles. The cell secretes certain molecules by the fusion of vesicles with the plasma membrane. This process is called exocytosis. A transport vesicles that has bubbed from the Golgi apparatus moves along a microtubule of the cytoskeleton to the plasma membrane. When the vesicle membrane and the plasma membrane come into contact, specific protein in both membrane uh, rearranged the lipid molecules of the two bilayers so that the two membrane fuse. The contents of the vesicle spill out of the cell, and the vesicle membrane becomes part of the plasma membrane. Many secretory cells use exocytosis to export products. For example, cells in the pancreas that make insulin secrete it into the extracellular fluid by exocytosis. In endocytosis, the cell takes in molecules and particulate matter by forming new vesicles from the plasma membrane. Although the proteins involved in the process are different, the events of endocytosis look like the reverse of exocytosis. There are three main or three types of endocytosis. We have phagocytosis, cellular eating, pino mean drinking, and phago mean eating. Cytosis of the cells, and we have receptor mediated endocytosis. In phagocytosis, a cell engulfs a particle by extending pseudopod, and singular is pseudopodium, around the particle, food particle, and packaging it within a membranous sac called the food vacuole. The particle will be digested after the food vacuole fuses with a lysosome containing hydrolytic enzymes. In pinocytosis, a cell continually gulps droplets of extracellular fluid into tiny vesicles formed by unfolding of the plasma membrane. In this way, the cell obtains molecules dissolved in droplets. In this intake, receptor-mediated endocytosis is a specialized type of pinocytosis that enables the cell to acquire a bulk quantities of specific substances, even though those substances may not be very concentrated in the extracellular fluid. Embedded in the plasma membrane are proteins with receptor sites exposed to extracellular fluid of the yellow Y shape. Those receptor specific solutes will bind to the receptors, and then the receptor proteins cluster in coated bits, and each coated bit forms a vesicle containing the bond molecules. Just like trap, those receptors attach with each molecules, and then they fold them. Those are serve as traps.